So thanks, Phil, for the introduction. It's a pleasure to introduce the Neil Bulleid integrated protein analysis, named in memory of Neil and the work he did within Glasgow University, but also in setting up an earlier incarnation of this facility. So just to give you a little bit of background, we started out quite a number of years ago now, offering limited range of methods, and it's sort of built up from there over time, using adding more equipment, adding more expertise to what we can actually do. Until in 2023, we were the structural biology and biophysical characterization facility embedded within the School of Molecular Biosciences. And simultaneously, there was also this protein production unit that was housed within the School of Infection and Immunity. As we then converged into the integrated protein analysis, the protein production joined us. So we're now a team of three. There's myself, I'm a structural biologist and biophysicist. There's June Southall, who's a technical specialist in biophysics, and there's Susan Kitson, who's a technical specialist in protein production. So I'll just briefly go through some of the things that we do and how they may be of use to you. So in terms of protein production, we offer bespoke expression and purification schemes based on your needs. So we can do E. coli, which is our favorite medium, of course, because it's easy, it's cheap, but we can also do insect and mammalian cell production if that's required. Most of the work we do is lab scale for downstream research applications. So lab scale can be anything from 100 microliters to, 10 mg per mil, several mils if you want to do something like protein crystallography or more advanced biophysical methods. But we're also working with local companies to try to scale up samples. So where we are doing the initial purification protocol, seeing if it's actually feasible, and then we're passing it on to companies that are more suited for pilot scale with 600 liters, and then where the end game is thousands of liters. And to do this, we try to stay in touch with the expertise in scaling up at an early point, as we can, to make sure that we're actually making sensible decisions throughout our part of things. There's no point in having a 7-step purification protocol and then trying to make that into an industrial procedure. And here's a couple of recent publications that have stemmed from our protein. So Susan has been overexpressing, purifying these proteins that has been used in crystal trials by and successfully yielded crystal structures by our collaborators at Strathclyde University. So once you have protein, we like to test that we actually know what it is. And we also see a lot of people coming in and doing this sort of quality assessment and general biophysical characterization of their samples. So we can look at several things. We can look at the folding of proteins and peptides. And this is particularly key if you're working on peptide and peptide mimetics, where the structure is actually essential. If you think it should be a small helix and it isn't, then it's not going to do what you think it is in your assays. So we're using circular dichroism for this. Basically, secondary structure components behave differently in polarized light, and we can then interpret these spectra of your sample and see, does the protein have a fold? Is it actually not folded at all? Is it disordered when it shouldn't be? Has it got the right? If you're comparing your wild type with a mutant, is it the same or has something drastically changed? And we can monitor that here. We can look at the correct molecular weight of your sample. By correct, I mean what it actually is rather than what you think it should be. And we can do that mostly via size exclusion chromatography with a multi-angle light scattering detector attached. And down here, I just demonstrate the results. It not only gives us the accurate weight of the sample, but it also tells us the ratios of the different peaks. So if you have stuff like this in here, this is aggregation coming up. And you're losing quite a bit of protein here, but more importantly, when that is in your sample and you're measuring, for example, the protein concentration, you're measuring the concentration of everything in here. So this is creating noise that you don't need. And you could also stop some of your further downstream experiments from working. So we introduced another purification step in here, or we said this isn't protein that we purified. This is a user coming in, and so we suggested they introduced another step, and we removed most of the aggregation here, and we got a bit of a cleaner peak of the correct size. There's still something going on because we have two peaks here. Not quite sure what it is, but it might be an incomplete cleavage of the his tag. Once you have protein and it isn't looking perfect, what can you do? Well, we can try to optimize the buffer conditions, and we do that using thermal shift assays, which allows us to determine the melting temperature of your protein in a given set of conditions. And we have a screen around 30 different buffer conditions, varying pH, varying salt concentrations. And we're looking to see the point when, or the conditions where your melting temperature has increased. So higher melting temperature means better protein stability, better solubility as well. So we're also developing a small molecule screen now that you can use as a secondary screen. If we find some improvement with pH and salt, we can also then start adding arginine, glutamate, glycerol, all that sort of thing to your sample and see if that can even further stabilize the protein. And we can elucidate how your protein behaves in solution. Currently, we're doing this via analytical ultracentrifugation. We're hoping to hear back from a BBSRC grant for mass photometry in the near future. And what analytical centrifugation tells you is how your protein behaves in a solution, if it's homogeneous or if it's got multiple populations. So what we're doing here, this is overlay of 5 different runs. And in the magenta line here, we see a protein. It's called BERK2. It doesn't really matter what it is, but it sits on its own, this nice population here. Similarly, SMAC, which is in blue, it's a different protein. It sits at the same size. So AUC measures things in Svedberg units, which is a way of giving size and mass information. So once you mix these two samples together, you see that we're actually in the cyan line here, we see we're actually getting a new peak coming up over here, suggesting that the two proteins are actually forming a complex that is moving much further along in the AUC system. And what we've done here then is we've tried to introduce SMAC to BERK2 in a ratio, and we see that we still have, when we have a 1-to-1 ratio, we have a peak here. So there's one of the two proteins that hasn't been reacting completely, and a peak here. We have 1-to-2, we have no peak in this area, and everything has shifted down here, suggesting that we're actually completed the reaction and we formed the complex. And then at 1-to-4, we're starting to get a second peak coming up again. So from this, we interpret this to mean that BERK and SMAC bind in a 1-to-2 ratio, and we can then confirm it later on using things like SECmass to look at correct mass or complete mass, and we can see that it's actually a 2-to-4 reaction. And whilst a lot of this is used early on in your stage, also some people are also publishing, this as part of understanding how their system works. So it's quite key. Biophysical understanding of your protein or of your sample is quite essential for many of your experiments. The next thing we can do is determine binding affinities. We have 3 methods at the moment. We have our high throughput option, which is SPR, surface plasmon resonance. You basically bind one protein or sample or whatever down to a chip, and then you can flood a range of samples across it, leaving it overnight. So you can have 96 single points if you want, or you can do 12 concentration curves overnight to get KDs determined from up to 5 different samples. For example, your wild type and a range of mutants. But one of the proteins is stuck to a chip. There's a completely label-free option, ITC, isothermal titration calorimetry. It's the gold standard in terms of determining binding affinities. It is quite greedy in terms of your amount of sample that you need. And it is essential that the buffers are identical. And I don't mean made up the same. I mean from the same bottle, or you will not get decent results from this. So, and it also, a run takes 3 or 4 hours. So this isn't the method you would use to start with, but it's a great orthogonal technique when you have your initial output from the SPR and then take 1 or 2 and you use ITC on that to finalize for your manuscript. If you have very small volumes, then microscale thermophoresis is a great way of looking at your sample. It does require fluorescent labeling. We have kits that can do that for you in the facility. But again, if you don't have much sample, this is probably where you would want to start. Together, these things give you binding affinities, KDs, they give you K on, K off, which is quite essential if you're trying to look for small molecules and drug development. You might want something that binds really quickly. You very often want something that stays on for a long time, so you don't want a super fast K off. What's the point in attaching to a receptor and then just disappearing again immediately? You can get things like changes in enthalpy and entropy. Again, people come in, they use the equipment. So here's some recent work from the Walden lab, and they're looking at the impact of ubiquitination on proteins and the binding affinities between them. So, sorry, my video is in the way and I can't actually move it. So basically what we're looking at here is the binding of 2 proteins, FANC-I and FANC-D2, on their own, or then if one of them has been ubiquitinated. And you can see that the binding association, binding affinity, increases dramatically once you have an ubiquitination in there. And this is using MST to identify. Similarly, Jesko Kanke, when he was at Glasgow University, again used MST to identify the Krogakin biosynthesis pathways and all the different components. So he identified here that Krogakin A and B interacts, A and E interacts, but A and C does not interact. And together with a lot of other work, obviously, then he could piece together the biosynthetic pathway that he'd identified. Finally, we do structural biology. We do high-resolution methods such as X-ray crystallography, protein NMR, and also in-depth structural analysis. So this figure here is all AlphaFold and AI derived. So you can stick in a sequence, you can stick in the SMILE sequence for a small molecule. It can come out with something that perhaps is useful. But the key is there's an overwhelming amount of structural data out there now, and we're offering help in actually finding the information in there that would help you. So if you're having an AlphaFold model from UniProt, we can help you with hypothesis generation. We can try to help you identify what are the sensible mutations you should make in order to see what it is you want to test. So come and have a chat with us if that's something that you would want some extra input in. But we also do low resolution methods, which I think is very interesting and very exciting, particularly when you have high resolution models that you can mix in with this. So we have solution angle X-ray scattering, also known as SAGS. And you can see here just an example. Here's a ubiquitin in blue, and then a single point mutation, residue 10 is mutated from a glycine to a valine. And you don't have to understand SAGS at all to see that there's something massively different going on here. And actually, if you then calculate surface envelopes, we see that, well, it's turned into a dimer all of a sudden. And then we've superposed it with a crystal structure, but we didn't actually need to here. You can see this almost by eye, what is going on. So SAGS tells you about things in solution that you might not be able to spot. AlphaFold wouldn't pick this up. Crystallography might not pick this up. So it's a very useful tool to look at dynamics and flexibility within a solution rather than at the still point. Similar, you can get information like that using, again, AUC, analytical ultracentrifugation, and we can do modeling. We don't get atomic resolution from this, but we do get domain-based resolution or sort of coarse-grained modeling from it. And again, here's an example that we published a couple of years ago now, where we have, as part of a much larger piece of work, we were looking for these virulence, antivirulence compounds, and we were identifying targets within E. coli as to what exactly these compounds did. We knew that if we gave these compounds to a bunch of 0157, so pathogenic E. coli, we would knock out the virulence and they wouldn't attack cells anymore. So we were trying to work out what are the actual targets. So this is actually the crystal structure of a spurious target. But we found the compound sitting here in the structure. We used this to try to identify a new and better set of compounds that had less off-target effects. And we succeeded in that. So the crystal structure doesn't have to be of, the one key protein that you're looking for, but this actually helped us identifying areas we needed to change in the small molecules to stop it from binding to, in this case, WRBA. And here's an example from the low end resolution where we were looking at the transcription factor EIF4A1 and how it binds to RNA and how that actually changes the overall size and shape of the protein. And this is very low resolution, of course, because it's a small angle X-ray scattering, but you can see that it's a sequence-specific change. So only in the AG-rich sequences do we see a big change and a sort of compactedness of the structure. And this was, again, part of a much larger piece of work, which confirmed that, but this was a key set of data for the initial part of the experiments. So what we can do for you, we can design expression constructs. We would like to speak to you as early as possible in any project. So if you're thinking, oh, I might express and purify some of that, come and have a chat. Tell us what you want to do with the sample afterwards, and we can try to design, a sensible expression construct. We might leave off some bits. You might use particular tags, etc. But involve us early on. And we can then purify that protein for you. You can come and learn how to do protein purification yourself. You can come and learn how to use all our equipment. Particularly students might want to come in, take part of collecting the data, learning how to use the equipment themselves, so that they can write it up as part of their thesis. We can quality assess your samples. We can tell you, does it have the mass that you think it should? Does it have the fold that you think it should? Is it as homogeneous as you think it should be? We can determine binding affinities to ligands or proteins of interest. We can do structural determinations at various levels of resolution, depending again on what is the question that you actually have. We can help you with analysis of computational predictions. We always write up reports, and we can help you with writing the appropriate parts for manuscripts. We provide quotes and supporting information for grant applications, and we're very happy to take part in joint grant applications as well, if that's something that you're interested in doing. And with that, here are my contact details and the general inquiries, and I'm happy to take any questions.